and therapy in Alzheimer's disease. And I am representing here Nensky Institute of Experimental Biology of Polish Academy of Science in Warsaw. And I am the leader of Laboratory of Preclinical Studies, as you could already hear in the introduction. And referring to what was already said, the disease that is described by Alois Alzheimer over 100 years ago is really the most common dementia worldwide. So over 70% of dementia cases is due to Alzheimer. Every 70 seconds, a new case is diagnosed. And we know that this is the dark side of a longer life because the risk for Alzheimer increases significantly with aging. You see here percentages. We will not go into these details. It's just the general picture. And this disease is the sixth leading cause of death and the third most expensive disease with tremendous global costs that affects not only the patients, but their families and the society. It represents a growing burden on the society and represents a great challenge for healthcare systems worldwide. Yes, it is the world's largest social crisis according to World Alzheimer Report because the number of AD patients increases. We expect to have in 2030 almost 70 million patients worldwide, and in Europe it will be around 15 million patients. This is the brain atrophy. So if we compare a healthy brain and Alzheimer's disease brain, we see the degeneration and death of neurons and shrinkage of the brain in particular areas such as cortex, so the area that governs cognitive functions, hippocampus, responsible for memory and spatial orientation, and in amygdala, in emotions. And in all these areas in Alzheimer's disease, we observed impairments in patients and in their daily functioning, daily living. We know that two main hallmarks of Alzheimer's disease in the brain, in neurons, are intraneuronal and extraneuronal deposits. So outside neurons, we see amyloid plaques composed of peptide, amyloid beta, whereas inside neurons, we observe neurofibrillary tangles, the structures, the deposits of hyperphosphorylated protein tau. And here I'm showing you drawings by Alzheimer that represent these structures in neurons as he observed. Yes, and we understand now, due to intense research in the past century, how these amyloid plaques and neurofibrillary tangles appear in the brain, how are they generated? And we know that amyloid plaques are formed due to creation of toxic amyloid beta peptide, and we know the enzymatic pathway that results in this generation of A-beta, so-called amyloidogenic proteolysis of the membranous precursor APP, amyloid protein precursor. And we also know the kinases, the enzymes that phosphorylate tau protein that makes this tau so toxic in our neurons. So, in other words, we know the therapeutic targets for Alzheimer's disease. And indeed, while this disease is very complicated, complex, and multifactorial, in fact, the main therapeutics that were tested in clinical trials for cure of Alzheimer's disease are based on amyloid cascades. So these are inhibitors of these enzymes, such as beta secretase and gamma secretase, that are responsible for generation of this toxic amyloid beta and generation of these plaques, as well as some other approaches, including tau. But in truth, the most efforts were concentrated on production and testing in clinical trials antibodies against this amyloid beta. So with the hope that they can remove toxic amyloid and improve cognition and memory in patients. And in fact, we witness several serious worldwide clinical trials with such antibodies. One of them, aducanumab, was registered and approved by FDA in states under accelerated approval pathway. And we have to admit, though, that this potentially disease-modifying therapy still did not provide proofs for cognitive improvements in patients. So this is a chance, and therefore the Centers for Medicare and Medicaid Services restricted Aduhelm coverage to clinical trials only. This treatment is associated with increased risk of cerebral edema and microhemorrhages. So in fact, we still lack effective cure for Alzheimer's disease. We have, of course, some treatments for symptoms, but we can't halt or cure the Alzheimer's disease nowadays. So lack of disease-modifying therapy. And here is again some illustration how intense these clinical trials and search for AD therapeutics are worldwide. This is the number of clinical trials conducted in many areas of the globe without, as to for today, without the effect, we still lack disease-modifying therapies on Alzheimer's disease. The research, though, help us to improve significantly diagnostics of Alzheimer's disease. And this is due to our understanding of this pathology, due to the research achievements, because now this diagnostic is based on biomarkers. So, of course, first it starts by assessment of symptoms with different neuropsychological tests, neurological assessment, and investigation of medical history. But from 2011, based on recommendations of the National Institute on Aging and Alzheimer's Association, we also support this assessment based on biomarkers, and these are biomarkers detected by brain imaging methods such as PET, and they rely on tracers that can detect amyloid beta, plaques, as well as tau with PET. There are also some other brain imaging tools such as MRI or particularly fluorodeoxyglucose PET that also measures structure and glucose metabolism in the brain that decreases in Alzheimer's. And in addition, we support the diagnostics on Alzheimer's disease with the assays of cerebrospinal fluid, again, amyloid beta and tau, total and phosphorylated. So these are biochemical changes that we measure in the CSF and that reflect the pathology in the brain, amyloid beta and tau pathology in the brain. And in fact, this introduction of biomarkers revolutionized the concept of Alzheimer's disease. First of all, we started to see these biomarkers and the changes in the brain already early at mild stage of Alzheimer's disease, as well as at so-called prodromal stage in mild cognitive impairment patients. Some of them with mild cognitive impairment, MCI, have Alzheimer's disease as a cause of this impairment, and some of them progress to further stages of Alzheimer's disease. So it's a great tool to be able to detect it in this stage. And moreover, we discovered that many cognitively normal So asymptomatic patients present in their brains deposits of amyloid beta and tau. So we discovered, particularly in this low percentage of patients that have Alzheimer's due to mutations, in fact, we are able to detect this disease 10, 20 years before the onset of symptoms, so before so-called clinical stage of the disease. And it's very important because it helps us to assess risk for development of Alzheimer's disease. And in our time, we witness the shift from symptom-based AD diagnostics to biomarker-based diagnostics, and we can even determine the stage of the disease based on the sequence of these markers' appearance in the brain, in the CSF. So we know that early diagnosis is a key factor for a better therapy. Of course, early detection allows us to introduce prevention, as well as the effects of drugs strongly depend on the stage when the disease is detected, because in fact, in Alzheimer's disease, we know that in later stages, the massive death of neurons give us no chance for effect of therapeutics, because the target of therapeutic doesn't exist anymore. So the earlier, the better for therapy of Alzheimer's disease. And in fact, what is particularly important is to be able to detect it in MCI patients. Usually this is nowadays the time when patients come to see a doctor to consult with the clinician. And moreover, we have a great need to detect it earlier at this asymptomatic stage. But current diagnostic methods have many limitations. Cerebrospinal fluid biomarkers require lumbar puncture, so an invasive procedure not suitable for repeated measures, for example, for monitoring effects of drug treatments. And brain imaging requires sophisticated equipment, is an expensive technique not available in many clinical settings worldwide. So what is desperately wanted in the field of Alzheimer's disease diagnostics are early biomarkers of Alzheimer's disease, preferably in blood, in easy accessible tissue. And in fact, these blood-based biomarkers fill the recommendations of the World Health Organization for a biomarker, a perfect biomarker, optimal biomarker. So they are present in readily available diagnostic tissue. The minimally invasive procedure is involved to detect them. And in fact, the assessment of these biomarkers is associated with application of not very cost-intensive assay methods. The application of biomarkers is much broader than the diagnostics only, because in fact, as I mentioned, they support us prognostics, detection of predementia, AD risk, and possibility of prevention, evaluation of response to treatment in clinical trials and later. And also, it is very needed for recruitment of patients for clinical trials of new therapies. Two paradigms are applied in the search for such blood-based biomarkers. First, we search for brain-derived molecules that could reflect brain changes, but also Alzheimer's disease is not only the disease of the brain. We see the changes in metabolism, insulin resistance is a sign and a risk for Alzheimer's disease, high blood pressure, and some dietary also, let's say, improper, unbalanced diet can lead to metabolic syndrome. That is also a high risk for Alzheimer's disease. So we search for peripheral molecular signage of AD. And to address these needs, the grant of European Union Horizon 2020 program was assigned to our consortium. This is a section on research and innovation actions entitled Novel Ideas for Radically New Technologies. And the grant title has a general topic, the screening, diagnostic, and therapy of Alzheimer's disease, with the acronym ARREST AD. So the mission is to stop AD, arrest AD. The coordinating country is France, and I'm very proud to say that out of 550 almost proposals, only 4% were selected for funding, and our project received the highest rating from the reviewers. So it also reflects how important the solutions for Alzheimer's disease are nowadays in the society. This ARREST-AD project is conducted and executed by the International ARREST-AD Consortium. The leader consortium is Professor Dulce Pape Garcia and her team at the Université in Paris, the Marne, in collaboration with the group of the Hospital de Paris. Professor Bruno Dubois is the head of this clinical group and collaborator of Professor Dulce Pape Garcia, but also our partners are in Spain, in Netherlands, in Great Britain, and we also have on board the company, the Screen Cell company. And I am representing Nensky Institute of Experimental Biology in Poland. So this is the picture from one of our meetings. We have regular report meetings with the leader of our consortium, Professor Dulce Pape, and Professor Bruno Dubois, and all the other members of this team. And what we try to do in this project is addressing the complexity of Alzheimer's disease. Alzheimer's disease and the causes is much more than A beta and phospho-tau. And we nowadays understand that there are changes preceding the deposition of amyloid and p-tau. So there are earlier causes that are addressed. And we need new hypotheses for these causes of AD to be tested, because the only one way for development of new drug targets and early biomarkers is the exploration of these causes. So this is the research for identification of new therapies and biomarkers that eventually result in neurodegeneration, as we know. So in ARREST-AD, we propose the early change in the form of a translocation in cells of so-called thiosulfated heparan sulfate, the glycobiological change, other words. So the exact title of this project reads thiosulfated heparan sulfate, translocation in altered membrane biology, a new strategy for early population screening and halting Alzheimer's neurodegeneration. So what this hypothesis assumes is that this translocation is the first change that triggers amyloid beta production and phosphorylation of tau, hyperphosphorylation, that eventually leads to neurodegeneration. We have the two arms, so one group of project leaders and their teams focuses on specific diagnostics in circulating blood cells, and the other group works on development of new therapeutics based on this heparan sulfate. And this is covered by European Patent Office, by the European patent. And right now, I will be not able to reveal the exact data, but I will show you part of them. We also study biological molecular mechanism that underlie the switch from pre-symptomatic to symptomatic AD due to these changes of glycobiology in neurons, in brain, as well as in blood cells in the periphery. Why this project is important? I think in the long term, we can provide a tool to reduce the risk of cognitive impairment and social exclusion of older people, increase their independence when we are able to arrest AD or cure AD, increase quality of life for caregivers, a serious issue in the field, and we also can support sustainability of health and care systems. We in the Nensky Institute, we are involved in diagnostic arm. This is my team. We are located in the Nensky Institute on the second floor, and we have a collaboration with the clinic, and this is the team from the so-called MSVA hospital, Ministry of Internal Affairs, and Mosakowski Medical Research Center, headed by Professor Maria Barcikowska, and the Alzheimer's division head is the Professor Tomasz Gabrielewicz. So we receive blood from this clinical team, and we collect this blood. We isolate human plasma in our task in this project in RSAD. So we receive human plasma from Poland, but also from France, due to the collaboration with our French partner, Professor Bruno Dubois and University Paris-Saclay. And we deposit this human plasma from Alzheimer's patients, mild cognitive impairment, also other dementia patients that are involved in this study. We compare these biomarkers and look for specificity for Alzheimer, and also from control healthy individuals in our biobank. In this biobank, we also deposit other cells from blood, cell lysates that we produce from the cells, and also nucleic acid that we extract from blood plasma and blood cells. We also have other resources in the biobank, but primarily what it represents is a unique collection of plasma and blood cells from AD patients. And having these resources, we are a member of Polish and International Biobanking Network, BBMRI. We are a member from 2018 when this network started to be constructed in Poland. And we are having audits that support us in also quality management and in maintaining generally quality standards in this biobank, including ethical and legal aspects, security, also information security, and general organization, institution management, so quality management in general. That is the basis allowing us to search for biomarkers of Alzheimer's disease in blood. First of all, we have to say that this is a particularly difficult task because of low concentration, extremely low concentration of brain-derived molecules in blood. presence of other markers released from blood cells and other organs that can interfere with tests, presence of endogenous heterophilic antibodies that interfere with many readings. So the fact that we can apply and use very sensitive, highly sensitive methods does not solve all the problems that exist when we try to measure molecules in blood coming from brain or small molecules also in the periphery. And many classes of molecules are tested as serum biomarkers. These are single molecules as well as multi-component panels that, as we think, can be more appropriate for multifactorial pathology, as Alzheimer's disease is a multifactorial pathology. And these classes of molecules include proteins, peptides, lipids, metabolites, low molecular weight compounds, and also a specific group of so-called microRNAs. So one arm of our biomarkers is on heparan sulfate as a biomarker, and the second is on non-coding microRNAs that we develop in the Nensky. What are these microRNAs? They belong to non-coding RNAs. And actually, nowadays, we understand that coding genes is only a small percentage of our genome. Majority of it, what makes us human also, what makes human brain a human brain, is this unimaginary ability for regulation of many proteins using these non-coding RNAs. So these microRNAs, they regulate 60% of gene transcripts, mRNAs. And there is over 2,500 these small sequences. These are only 22 nucleotides long fragments of RNA that regulate majority of gene transcripts. They are deposited in the database. We can identify them, and they bind to transcripts in so-called 3' UTR and block translation. So block production of protein from this RNA molecule, and they form a complex regulatory network and block and not only the translation, but also they cause degradation of these transcripts in complex protein systems, protein complex known as RNA interference, RNA-inducing silencing complex responsible for so-called RNA interference. This discovery was awarded in 2006 with Nobel Prize in Physiology and Medicine because it really affects regulation of many proteins in our body and in our brain particularly. There is a high number of microRNAs in the brain, as I mentioned. Okay, in addition to intracellular functions, these microRNAs are secreted, so they can function extracellularly. They can be produced and released from cells to the bloodstream. And in the bloodstream, they are transported and they can affect distant cells, so they can function similarly to hormones. And therefore, they are very promising as Alzheimer's disease biomarkers and in general as blood-based biomarkers because they are released from cells protected from degradation or in membranous vesicles or in protein or lipid complexes. So therefore, they are abundant and very stable in blood. Moreover, these vesicles, called exosomes, can cross the blood-brain barrier, so they participate in so-called brain-blood crosstalk. They can also mediate this crosstalk with other organs and in blood. And also they show tissue-specific expression. There are brain-specific microRNAs. All of these drags a lot of attention of researchers worldwide to this microRNA group in blood, so-called circulating microRNA. So we did a review of all this literature, and we found 34 studies in the decade 2007-2017 that reported 137 differential microRNAs in AD blood, but no consensus microRNA has been identified because of methodological variability. And in addition to this lack of standardization of protocols, we identify lack of studies in this particular early group of mild cognitive impairment patients that are experiencing these cognitive impairments due to Alzheimer's disease, prodromal Alzheimer's disease. So we address this need, and the novelty and uniqueness of our study comes from the fact that we applied standardized procedures recommended by European Union Joint Programming for Neurodegenerative Diseases. This was a big project, including consortia in Europe, Biomark APD, and we based the recruitment of patients not only on symptoms, but as recommended on CSF biomarkers. And we received plasma from these early AD patients, MCI due to AD. And in fact, we identified first microRNA biomarker panel in early AD blood. We patented this panel. These were 6 core microRNAs, but in total we identified 15 significantly altered microRNAs. And we also described it and developed it in several publications starting from 2017 till now. And what this panel detects is it detects in blood the Alzheimer's disease in mild cognitive impairment, and moreover, now in frame within this ARREST-AD continuation of this research. ARREST-AD supported this research, and we received as I mentioned, plasma from patients from Paris and from Poland. And in this group, we demonstrated, we had a very unique group of these patients asymptomatic at risk. These patients had the diagnostics or indication of the presence of amyloid beta in the brain, but they had no symptoms of any cognitive impairment yet, so they were asymptomatic at risk. And here in this group, we also verified these blood-based biomarkers. So this panel detects pre-symptomatic Alzheimer's disease. How we arrived at it? Yes, first we started from almost 200 microRNAs, the so-called microRNA panel that we develop as a plasma panel, and here we found in this pilot study in plasma based on so-called real-time quantitative PCR, these 15 microRNAs, 6 best, 9 novel, and 6 previously reported. You can see here this ANOVA heat map, and patients in MCI or AD had different pattern than controls, obviously. This is a fold change for these 15 microRNAs. 6 of them were most significantly altered, so they are particularly important. Here you can see it in green. And we verified it not only in this first pilot study in frame of Biomark APD grant, but also in Polish NCN, National Science Center grant, Opus, that allowed us to check it in an independent cohort of patients, 63 patients from Białystok and from Warsaw, and as I mentioned, in French and a new cohort from Poland in Arrest AD grant. So it referred to over 100 patients, Polish and French cohort. And these were blind tests. This microRNA profile is similar in MCI patients due to AD and also in early AD. We checked that this biomarker panel, microRNA panel, has high specificity and sensitivity for AD diagnostics. This is a mathematical equation. It is very high. The highest is 1, usually is not achieved from amyloid beta, for example, in the CSF. It is about 80% or 0.8. Over 0.8 here, it is even higher for particular microRNAs of our panel. And moreover, we started to analyze the function of these proteins, first using bioinformatic tools, and also we verified this function experimentally. What was striking to us was that these microRNAs regulate proteins known to be involved in pathogenesis of Alzheimer's disease, so this precursor of amyloid APP, the enzymes that produce amyloid beta, also this kinases that phosphorylate tau, but also they regulate proteins described in so-called non-amyloid hypotheses. There are mitochondrial proteins, apoptotic proteins, insulin-resistant related proteins, and oxidative stress. And in fact, they multiply the effect by targeting several same proteins. So we observe a very interesting synergistic regulation. It's very convincing to us that it is really deeply involved in pathogenesis, in pathomechanism of Alzheimer's disease. And based on this notion, I can say that we had another idea. So if each of these microRNAs can signal impairment in particular signaling pathway or particular proteins involved in Alzheimer's disease, it means that we can really use this molecular signature for the readout of for a readout of signaling pathways that are participating in particular patients in Alzheimer's disease. This would significantly improve, for example, the clinical trials with new drugs, because if we know, for example, that in patient 1, we observe that this microRNA is highly deregulated and it signals us the regulation of this enzyme that produces A beta, we know that for these patients, the preferable treatment is with inhibitors of this enzyme. And not for this patient. For this patient, the preferable one will be, for example, the treatment with inhibitors of phosphorylation of tau, because we could read it from the level of this microRNA. So we think that, and we develop this concept further to use it for individualized, personalized therapy, for proposing the more specific for a particular, as we call it, endophenotype of the disease, for better treatment options and better selection of patients for a given treatment. So, conclusions. We identified and verified a microRNA panel as blood biomarkers for prodromal early Alzheimer's disease, MCI, and as we think now, pre-symptomatic as well. Interestingly, we found that this microRNAs panel regulate signaling pathway related to Alzheimer's disease pathogenesis, and therefore we think that this panel could aid selection of individualized therapy. And with this, I think that this approach in diagnostics can indeed revolutionize the diagnostic as well as therapy by adding to it more specific approach. Yes, I would like to acknowledge my team that was involved in this research, as well as funding institutions. We did it, as I mentioned, based on several European grants, as well as a National Science Center grant, Polish grant, and we hope to continue this collaboration further, aiming to arrest this terrible disease. Thank you very much for attention, and I can be happy to respond to any questions that you may have.
Thank you very much, Professor Wojda, for this fascinating lecture. Also, first of all, congratulations on the position of the Polish science and your team in this project. It's very important for us to, first of all, know about this and then talk about this and be proud. I have two questions just to start the conversation, and then we'll ask the audience to pose questions as well. If you look at the history of the research in Alzheimer's, what would be the turning point that made this research and all these exploration really different and gave some hope for the patients?
It's a very good and, in fact, a deep question. I see it as a continuum rather than a breakthrough. I see it more as a intense effort of a network of scientists working on Alzheimer's disease and becoming more and more aware about the complexity of it. Maybe I will briefly show you. Sorry. I think I have this slide. As far as I understand, yes, you could hope see it. And now, this disease is very complex and multifactorial. I had this slide as an additional one and remove it, not to make it very complicated. But in fact, we step by step, we started from the discovery of amyloid and the generation of amyloid in the brain. This is, of course, a milestone in the discovery of this pathology and has tremendous impact on progress in diagnostics as well as therapeutic trials. So I think later this line of research that identified here the proteins that phosphorylate tau, locations, mechanism in the brain, and the effects of these two main factors in pathology. But later, step by step, we understood what is now very obvious, how much these processes contribute to neuroinflammation in the brain. I mentioned bad diet, a lack of physical activity, very well-known factors that affect metabolism and disruption of this metabolic homeostasis in our brain that affects brains. microbiota, we know now. And this all contributes to neuroinflammation in the brain. Neuroinflammation drives appearing of A-beta. This homeostasis in all the regulatory processes of proteins, removal of these lesions from the brain, degradation of these two bad boys from the brain, oxidative stress, insulin resistance. So I have to say that this was a continuous progress, as I said, and also it is reflected in the fact that right now we redefined Alzheimer's disease. It is no longer understood as a psychological phenomena. We understand it now as a biological continuum. So we started to indeed see the many aspects of progression and the aspect of our brain capacity to inhibit this progression. This is what we try to support as well. Yes, I think that that the division of disease as a continuum and research as a continuum, in this case, is the best description of where we are now.
Thank you very much. It's very helpful. And now I would like to actually jump from the molecular level to the population level. And what would be the factors that are actually accelerating the process, whether, because we have more and more cases reported, whether this is a question of reporting more cases or really increase in incidence and prevalence of this condition in human populations, and maybe there is some difference between different countries and regions and continents.
Yes, this knowledge is available. The statistics are provided on common fora for Alzheimer's disease. Alzheimer's disease report provides these values, and therefore we can say that yes, both factors you mentioned, so the frequency and availability of diagnostic tools makes the diagnosis of Alzheimer's disease more often. That's one aspect for sure. But in addition to it, we see the increase in number of patients. That is due to, I would say, the factors that affect aging population particularly. And these are many factors. Toxicity of the environment. There are many reports on it. The bad habits related to diet, lack of physical activity. Physical activity particularly affects the brain, not only due to metabolism, but also by inducing production of neuron growth factors in the brain. So the stimulatory of our neurons. Definitely, there are list of risk factors for Alzheimer's disease. Infections. We know that bacteria that can invade the brain can come from tooth disease, for example. We know that the COVID-19 pandemic demonstrated that viral entry to the brain can affect brain. We knew it before because of studies on AIDS and dementia following AIDS disease. So definitely we see that there are a variety of damaging factors that can affect the brain and that can induce gradually neuroinflammation. I would say nowadays we see these hypotheses as a driving force for Alzheimer's disease, but Alzheimer's disease can develop not only due to one type of impairment. There are different types of impairment, and also, as we mentioned, a lot of significance is related to our brain capacity to respond to it, so our resistance to it. That's why it is so difficult to prognose progression of Alzheimer's disease. But there are, of course, data showing that we can eventually arrive to it. So yes, two factors. First, the increase in all the technological availability for diagnostics, more frequent diagnostic of Alzheimer, and I would say the factors of our civilization that increase the rate of Alzheimer, not only in demographically aging populations, so very advanced populations, but also in poor countries. So it shows that also the stress coming from the environment is a significant factor in this growth.
Thank you very much for this very specific answer on the molecular level and population level. I would like to invite my co-moderator, whether she would like to ask any questions. Professor Hutin, please.
Much for your very interesting talk. You answered partly one of my questions. You say it's very difficult to prognose Alzheimer's disease, because I was wondering if your research with the disease continuum research steps would help also not only diagnostic tests, but also the prognosis space in order to prevent even more detecting very, very early stages. So you said it's difficult because of all these risk factors and bad lifestyles, but on the other hand, you did a lot of research already on all these risk factors. So is there any hope that it can also help prognosis space?
Yes, and already in addition to microRNAs, of course, it is my field. We have now this confirmation that this molecular signature is present in early stages, these pre-dementia stages. So yes, indeed, they could be further developed as prognostic biomarkers, not only diagnostic biomarkers. We also know from the breakthrough publications in 2021 and 20 on phosphorylated tau detected in blood by a very sensitive methodology that is also detected very early in pre-dementia stages, and it's also a hope for becoming a prognostic biomarker. So yes, we already see this prognosis. Now, important issue is how to mathematically provide the most specific information. How big is the risk? How to calculate the risk rate based on what we see. The signature is there, but to indicate when we can expect the development of the disease in a given patient. Is this 10 years? Is this 3 years? Is this, how probable is this development? So I would say here is the issue, but the general findings, the basic findings that the signature is present in pre-dementia stages is already done.
Question is, you said you use marker released in blood cell, but on the other hand, you said that there's a low concentration of brain-derived molecule in blood cells. So can you, what are the conditions that ensure that your test is going to be reliable and for populations?
Mm-hmm. Very good, again, very good aspect and very good question. Yes, the whole study that we did was based on quantitative real-time PCR. That is methodology that allows amplification on microRNAs in plasma, because of course they are very, very diluted. The general concept is that we see brain-derived microRNAs that naturally cross the blood-brain barrier and are present in bloodstream. So this is different from amyloid or tau that are in fact more diluted, let's say, in blood than microRNAs that are designed to travel, maybe. That's what we are guessing, to communicate with periphery. That's one thing. But in addition to it, we tried several techniques, and the lack of this repeatability that I mentioned in the previous studies comes from the fact that people use different methods, and we lacked standardization of these methods. So first of all, using different techniques, we show, this is the article that we are working on now, that the individual microRNA panel, not the high throughput study that can be normalized to a mean level of all of these microRNAs, but individual single molecules in the panel, they require absolutely the amplification step. So this is the power of the methodologies that we are providing, that it amplifies significantly the molecules. Now, if we are using such a technique, we need to control this amplification step very clearly. So we are preparing the recommendations for the PCR methodology based on several controls, double controls for each step: efficiency of isolation, efficiency of amplification, and so on. And moreover, we verified a set of normalizers for their stability in aging population. It also was not done previously. So we are providing methodological tools to support this assessment in a reliable manner, in comparable manner. Without it, we will not be able to move to diagnostics, basically.
Thank you for this great presentation. I have a question. It's more on the higher level. I guess you mentioned the complexity of the Alzheimer's disease, and it's connected with neuroinflammation, also like exposure of certain proteins. But there was a drug that was introduced by Biogen, and it didn't actually had a really bad public opinion just because it didn't work. So how did it affect your fields, or what do you think about introducing such novel drugs to the market, but they are not well tested just because it's also really hard disease? And that, I think that's the first question. And the second question, there's also lots of hype, for instance, then lack of sleep can increase the chance of Alzheimer's. Is it true? Or also like apparently smoking can decrease risk of Alzheimer's. Is it true as well?
Mm-hmm. Maybe I'll start from the second question. As far as sleeps and time of sleep goes, yes, there is a body of literature that documents this effect. And it is well understood at molecular level. Our brain needs the night rest also to remove the deposits of amyloid, deposits of other aggregates from the brain. And the minimum timing for it is about 6 hours, as it is generally described. So the difficulties in or lack of sleep affects these processes of brain removal of all these toxic molecules. This is one of I think, well-known effects of sleep right now in the field. The smoking. I would say that, of course, there are different studies. These are population-based research, basically. However, despite that, we think about a nicotinic receptor in the brain that could be, activated. But in fact, the population-based research in meta-analysis of high number of patients in cohorts shows that nicotine addiction and smoking is a risk factor for Alzheimer's disease. So it gives more harm than good. Now moving to drugs. If we go to Alz Forum, the webpage for Alzheimer's disease, we can see a very hot discussion on this decision of FDA to introduce the first antibody against amyloid beta. This was done based on the proof that this antibody really removes the amyloid plaques from brain. But the removal of amyloid plaques does not improve, so far, we do not have any proof, the cognition or memory in patients. So this was introduced with the hope that this decision will help researchers to move forward in verification of this drug. However, it also is broadly disputed as to lack of this proof. So EMA in Europe, for example, did not approve this drug, right? There are, of course, there is some value in the introduction, because one of the serious disease factors is removed from brain. So we can await maybe a beneficial effect in a long run. On the other hand, why it is so disputed by many medical doctors and researchers is that if we understand the Alzheimer's disease as a very complex dyshomeostasis in brain, maybe removal of one factor, even very important, such as amyloid plaque, will act as stopping smoking in a patient who developed lung cancer, right? The cellular homeostasis is already at different level, and too many other factors are playing role in pathology. So the removal of one will not solve everything. It also shows that we need multi-targeting approach in Alzheimer's disease, I would say. But at the present moment, the data are collected. So time will show whether this was the right decision or not.
Thank you very much. You really get us thinking right now. I would like to see if there are any other questions in the audience. Dr. Woźniewicz?
Dzień dobry. Thank you very much for this very interesting lecture. Of course. So I have just a quick question. There are also markers via QEEG for Alzheimer, and I would like to ask, do you compare for the same patient when you already have your patients the result from blood and from brain? Or you think EEG?
Yes, this is an obvious study to do because all our patients are diagnosed based on biomarkers. So they have very solid documentation based on symptoms or on all classical aspects. A group from France received amyloid PET data, whereas patients in the cohort from Warsaw Hospital and from Białystok, it's another cohort, they had CSF markers. So they have amyloid beta and tau markers. And what we do correlation studies between these markers and our microRNAs in MCI due to AD because of this importance of this group as well as in asymptomatic. I would say that individual biomarkers are showing that some of them are more related to amyloid, some are more related to tau. And it is, I think, understandable because of a variety of pathologies that contribute to this disease. But some are unrelated. Nevertheless, we can say that a panel as a whole is related. Of course, we receive all these patients that show these changes at the level of amyloid and tau. So, in summary, I would say yes, some of the particular microRNAs show correlation with other known biomarkers, and some are novel. They report other aspects of Alzheimer, and they do not show correlation. A panel as a whole panel, yes, it is related.
Thank you very much. And if we have time, just very, very short question. Is there any moment you would say which is like early enough to have the treatment or the moment which is like too late for whatever you are working on? Because as we know, a lot of people which has dementia symptoms, at the beginning, they just ignore it or sometimes they just go to doctor too late, or is there a moment you could catch from whatever you're doing now?
This is the experience that is coming, I would say, mainly from many colleagues working in clinical trials. For us, we have to be humble and be very cautious with any, recommendations or preventing anybody from trying therapy. I would not be in this position from obvious reasons. But in fact, we know that many clinical trials failed because of wrong recruitment of patients that already had too high degree of neurons damage in the brain. So in fact, it is a danger that starting contacts with clinicians late in this disease may be really detrimental. But how early you should start, I think the earlier the better. And in fact, there is an ethical issue because right now we can't offer a cure for this disease, right? We can offer a symptomatic treatment that can support these patients in this stage, and antipsychotic drugs, some, mainly they are inhibitors of acetylcholine esterase that block symptoms related to lack of neurotransmitter in the brain, acetylcholine. But in fact, these are symptoms only. So the issue is what we are providing patients with, and we inform about the risk of the disease. But patients themselves, it gives them possibility to change their lifestyle, make a significant decision about the aspects of their functioning, And of course, even information about the disease earlier allows symptomatic treatment to be introduced earlier. So I think it is beneficial in many ways.
Thank you very much. We hope that this is just the beginning of our communication, and we'll be in touch, I hope. And thank you again for sharing with us this groundbreaking and very, very new information. We'll continue our discussion. Right now, for the sake of time, we have to move to another session. And thank you again. Thank you so much.